Assalamualaikum and very good morning. And if you remember back in the day when we were little, our grandparents used to boil bitter leaf and use it to treat for most of our problems, from skin related problem to even stomach ache. Yes, my proposal is about the genus from where the beetle belong to, which is piper. Not knowing hence not living. A little bit background information about this plant is that it is a pantropic species, which means it can be found in tropical region of the earth. They also have been known to be used as spice, ornamental, and even as an alternative treatment not only in Malaysia but around the world. In this research, we are going to specifically study about the secondary metabolite, which in simple terms means an organic compound that's not directly involved in the growth of plant, and the absence of it does not directly kill the plant. These metabolites are also known to be used by the plant as defense mechanisms against disease-causing microorganisms, and humans had known to utilize them to use in industry as flavoring, coloring, and also as medicine. To further strengthen and support our research about the metabolite from this species, we had collectively extracted some publications to help us fully understand about this plant. From that, we have found out that these plants are rich in metabolites, such as the study done by memorandum shown that these plants have alkaloid, phenol, flavonoid, tannin, saponins, glycoside, terpenone, and also steroids. And also deeper research about the alkaloids of the plant shown that these have phytolate, acyclic ditrophin alcoholics, 4-chromanol, and hydroxychavicol, which are deemed to be the ones responsible for their antibacterial properties. We also have found some application on the effect of this species toward two most famous pathogens. The first one on bacteria done by Vet et al. The studies was against several aquatic bacteria species, and the extract of piper have positive emission on all of the bacterial species. And what's more interesting is the studies done by Severedy, which the ethyl acetate extract of the piper are deemed to have an inhibition effect greater than fluconazole, which is the used common prescribed drug. We also had found some publication that these species have antioxidant too, which done by Insano shown that 4 piper species and 4 of them give positive feedback, with piper nigrum has the greatest value. From the information that we extract, it is clearly stated that these plants are very promising in terms of their richness and also their beneficial effects. Our main question is which part of the plant are responsible for their vegetative activities. So in order to fully understand that, we are going to conduct an isolation and characterization of the secondary metabolite from the species, and the isolated secondary metabolite will be tested for their biological activities. From that, we had proposed a method with some modification. Science is beautiful. It does not need to be complex, but the most important thing, it has to be effective. So in our experiment, we are going to use the thin layer chromatography technique paired with gas chromatography and mass spectrometry for further education of the secondary metabolites. We are going to choose 3 species and they are going to be prepared in an ethanolized track. After the sample had been completely isolated, we are going to use the TLC chromatogram and test using 2 different assays, which is the agar-Overlay assay and the TLC contact assay, which we are going to test on 3 different pathogens, which is the gram-positive, gram-negative, and the fungal species. Their antioxidant assay are also been studied by using 2 different methods, which is the total antioxidant capacity and hydrogen peroxide scavenging. From our research, we expected that all 3 species to inhibit the growth of all 3 microbes, which piper beetle, are expected to have the greatest inhibition factor. Also, the effectiveness of the antioxidant of the piper is also predicted to have a very good antioxidant properties. This is our work plan, which consists of data collection, preparation of plant sample, TSC and GC for isolation, and also the biological tests. We also want to thank some journal that really help us in more understanding about the plant and their metabolites, which we include in the reference. Piper are one of the most common species used as medicine in the past, and with my proper research, we can fully utilize parts of the plant. In conclusion, piper species are very promising for future studies and can be used as future medicine. Thanks.